Good evening, everyone. Today we'll be talking about the evolution of the human gut microbiome. Forgive me for seeing the presentation part of this. I've been trying to figure it out, but it was not really working. So, um, first, we're going to start off with uses of bacteria. And so we know that bacteria is being used every day for certain things. For example, it assists in food production with cheese, yogurt, ice cream. It also produces antibiotics and probiotics in the healthcare industry. It helps clean up oil spills and also converts waste into fuels. But most importantly, what we're going to talk about today is how it helps uh, human life by presiding in the gut. So we have bacteria in our body. And for some people, this may be new. For most people, it's not. However, it is something that's a little bit unknown, or not unknown. It's unknown the cause or the effect of that it has in our body. So we can see here that it ranges throughout the whole GI tract, the esophagus, through the oral cavity, the esophagus, stomach, small intestine, large intestine. So the types of bacteria vary depending on the pH because the pH is so varied in the body. So here in the stomach, we have a high pH. So there'll be different types of bacteria compared to that of the small intestine or the esophagus because that's a much more neutral pH. So less or more acidic in the stomach and less everywhere else. Most of the bacteria in the body resides in the colon. It's 10 to the 11th power bacteria that reside in the colon, and that's where the most diverse areas or the most diverse colonies of bacteria live. So these are the types of taxa in the body. So we can see humans, mouse, rats, and pigs. Firmicutes and Bacteroidetes are within all of them, but we have actinobacteria and proteobacteria as well within humans. We're not going to talk about archaeoviruses or eukarya. There's probably much more research being done on those, but bacteria is what we're going to focus on today. So there's much diversity of bacteria in the body, and this is a study that was done where it showed this diversity. So what researchers did was they took distal gut-derived 16 srRNA sequences and made a phylogenetic tree of them in mice and humans, and then put them together. So humans are blue, mice are red, and we were able to see that there's over almost 12,000 rsRNA sequences and approximately 5,000 mice srRNA sequences. So if we look down here, Bacteroidetes resides in both species is very diverged. There's a big part for mice and there's a big part for humans, and they're very branched. But we get up into here into actinobacteria and proteobacteria, and there's mostly blue, as we saw earlier, as we talked about here. Those mostly reside in humans. So we can see this branching and divergence of different types of bacterial species inside this living species itself. So now we're going to look at the biogeographical diversity of the gut, and then also this. Well, here we'll look at the species diversity of the gut, and then we'll look at the biogeographical diversity within humans specifically. But here we can see that there's different types of gut microbiomes, and they're within different types of animals. For example, fish, reptiles, bats, birds, etc. So this is all the differentiation, whereas like fish are totally opposite from reptiles, and mammals are totally opposite from bats and birds. Now, if we look here, this is the most important and to me, this is what really separates why we have such different gut microbiomes as humans. So this is looking at the biogeographical diversity of the gut microbiome, and we're taking samples from the United States, Venezuela, Malawi, which is in East Africa, and Yanomami, which I don't know where it's at. So the United States obviously is a very urban area, whereas these other areas are more rural. And we can see that these rural areas are much more differentiated than the ones that are within the United States. So, excuse the background noise. I'm at work right now. I'm on call tonight. Anyways, why are these so different and diverged? Why are they so separate? Now we can look at the the dieting habits of those humans living in these urban areas compared to rural areas. Those who live in urban areas live or eat mostly sugar and protein diets. That's the majority of what they eat. Carbohydrate diets, high carbs, and high protein. Whereas those living in rural areas eat mostly high fiber diets, which is really important. That's mostly where we see huge proliferation of bacteria with these fibrous diets. That's what bacteria like to eat. That's how they become to be more diverse and such. So we see that bigger diversity of those high fiber diet areas compared to the high protein, high sugar rural area or urban areas. So if we look at this graph, this also explains what we see. We can see here as humans in urban areas taken from the United States and Italy. They're they're not, there's not much divergence or diversity. There's just kind of the same species. Whereas you get that high fiber intake, Tanzania, Malawi, Peru, Venezuela, and you get this huge diverse separation of human gut bacteria, which proliferates because of that fiber. There's more species able to be found and such. So now we can take this into medicine, and specifically with autism spectrum disorder. Now, children with autism spectrum disorder suffer obviously from neurological problems, but these neurological problems also cause behavioral problems, which social ineptness and stuff like that, but also in the types of food they eat. Children with autism spectrum disorder typically exhibit eating habits which are based on texture, color, taste of foods. And most of these foods are junk food. They like foods that are sugary, uh, high carbs, high gluten or high fructose corn syrup. Like I said, junk food, candy, cereal, cupcakes, gushers, fruit roll-ups, you name it. Those are what they'll eat, and if they don't get what they want, it's just there's that social ineptness that comes to play. And because of this, they're very limited in what types of bacteria can reside in their gut. And that's what these research these types of research is trying to show. So these are just different studies that have been done. I'm going to talk about the top one and the bottom one. But children, or first we'll look at the top one, autism spectrum disorder animal model. So these are rats that display autism spectrum disorder. They were given a probiotic, and they improved gut barrier integrity, normalized gut microbiota, and reversed autism spectrum disorder related behaviors. Now that's interesting. They reversed the behaviors that the rats displayed in regards to autism spectrum disorder. That's incredible. And this treatment was done in 2013, so obviously it's very recent. Now, if we try this, they tried this with humans, and it's not the same type of treatment, but they did a little bit different. As we'll see, they had 18 children with autism spectrum disorder. They did a microbiota transfer therapy, 14 days vancomycin treatment, a bowel cleanse, and administered a high initial dose of standardized human gut microbiota, which is taking the human gut microbiota out of one person and putting it in the other. They did it for 7 to 8 weeks with the human gut microbiome, with 2.5 times 10 to the 12 cells per day. This improved both their gut GI symptoms, constipation, diarrhea, indigestion, abdominal pain, and their autism spectrum disorder related symptoms. So that's not technically talking about behaviors. However, it could, if we figure out the perfect treatment, this could change what we see and hopefully come up to this, help us relate more to this model with autism spectrum disorder related behaviors being reversed. So this is kind of the breaking boundary here with the human gut microbiome. I hope you learned a lot. I hope you're able to see how much our diet affects the change and the diversity of our gut microbiome and how important it is in our body. Here's some of my references I used. I used a lot of these in my paper. Like I said, I hope you learned much, and thank you so much for listening to my presentation.